Today we're going to be looking at populations. Populations is actually a topic that you're going to learn about in biology, so this will come back next year. Now, we've already talked about what a population is when we looked at our biodiversity unit, but just as a review, it's all the individuals of a species that live together in an area. So it is one particular species. When we look at populations, we're not talking about the number of every single species within the area. That would be a community. We are looking at one species that lives together in the same place. So one species, same place at the same time. That's what we're looking at when we're defining our population. As we look at populations, there's three main things that we look at. We look at the size of the population. So that's going to talk about your numbers. We're going to look at density. So how many individuals per area of space, because when you think about density equals mass divided by volume. That was our formula. Mass is the matter, so how much stuff, and then volume is the amount of space. So specifically when we're looking at it here, we're going to look at how many of that particular species within that one particular area of space. And then dispersion is going to be more of how they are spaced out. And we'll look at those each individually. So size. Size is the number of individuals in an area. So what is the population of this particular spot right here of these swans? Well, the population would be 2 because there are 2 swans, in fact, standing there. Now, again, remember, when you look at the size, we are only talking about that particular species. So if I'm going to find out the size of population of ants in my backyard, I'm only going to look at ants. I'm not going to look at the spiders and whatever else is back there. So size is pretty simple. And then later we'll look at how we count that. So then your growth rate. This is what's going to affect the size of your population or determine the size. You have your birth rate minus your death rate equals your growth rate. So how many individuals are born versus how many individuals die. When you're looking at the size of your population, you are constantly adding more species into that population by the birth rate. So that's going to be adding. And then your death rate is subtracting. And that's what together creates that growth rate. You can't just look at birth rate because if you have more people who are dying than being born, even though you still have individuals being born into the population, your population as a total is going down if your death rate is higher. Now, I don't mean for this to be a sad story, but when my sister had her baby, on the same day she had her baby, my brother-in-law's grandmother died. So the birth rate would be 1, the death rate would be 1, so the growth rate equaled 0. Even though there was a birth, the population did not grow at all because there was also a loss on that same day. This is what we call down here an age structure. We're going to look at this a lot more, but it's basically a graph that focuses on how many individuals are within a particular age. And we will come back to that in just a little bit. Now, when you look at density, I already reminded you that density is mass divided by volume, but again, that's what we have here. You have mass, which is the number of individuals of the same species, divided by a unit of space, which aka is your volume. So it's just how many of that particular population is within that area. If I have 6 people within my classroom, then my classroom is not very dense. If I add 30 people into my classroom, it is now very dense. And you may think, well, why do we even care about the density of a population? What does that affect? Well, that actually affects a lot because that gets into overpopulation and some other factors that we'll talk about in the next slide or two. So some other things that can affect density are going to be things that can also affect the growth rate because the growth rate is going to be directly related to the density. You have, and these are really just vocabulary words, but you have immigration. Immigration is movement into a population. So when you're looking at in, it starts with an I, and immigration also starts with an I. So if you are immigrating somewhere, you are moving into that population. So that's going to cause your population to grow. So I'm going to put a plus sign there. Emigration is when individuals exit or they leave. So I'm going to write exit to the side. So immigration, I like to say it so that the E is kind of emphasized, but immigration exit. And that will help you remember the difference between immigration in, immigration out. So that's going to be a minus. That would make your population smaller. Then we have what we call density-dependent factors. I want you to think back to Unit 1. In Unit 1, we talked about a independent variable versus a dependent variable. The independent variable was what you changed. I changed independent variable. And it's that one thing that you were causing to change within the experiment. And then a dependent variable changed depending on what you did or as a result of what you did. So when we are looking at a density-dependent density-dependent factor, we are looking at something that depends on the density. So this depends on what the density is in terms of how big of an effect it's going to have on the population. These types of things are typically biotic. Remember, bio means life, so biotic factors are living things. And so these living things have a larger effect on the population as the size gets bigger because that means the density goes up. An example to help you understand this a little bit more is going to be something like disease. This actually goes really well with the whole COVID stuff that's been going on. So with COVID, you have more of an effect in your higher populated areas. So as your density goes up, that means the amount of disease that's spread is also going to go up. Because think about it, if you are in New York and you have a subway full of people, if one person is sick on that subway, they're in very close contact to everybody else, so it's going to spread much easier. Another example is going to be something like competition. The more people you have, the more competition there is. Think about if I have a pizza and I've got 5 slices of pizza. Well, if I only have 2 people in my room, there's not going to be a lot of competition over the pizza because my density is very low. My population size is low within my room. But if my density increases and my room is now filled with 30 people and I only have 5 slices of pizza, then the competition is going to go way up. So these density-dependent factors have a much higher effect on the population as the density goes up because the effect of the factors depends on the density, which is why they're called density-dependent. So I'm going to clear my screen and we'll go on to density-independent factors. Density-independent factors are factors that are going to affect the population independent or regardless, I'll write that out, regardless of what the population is or what the density is. So these are typically abiotic factors, and they, again, are going to affect it regardless of the density. So temperature or water. If you have a lack of water, every species needs water in order to survive. So let's say you have a forest that has 10 little squirrels in it. So it is not a very dense population. Well, if you run out of water, then all the squirrels are going to end up dying. They won't be able to live there. They're either going to have to die or they're going to have to emigrate out of that area. If you have a very dense population where you have 50 squirrels in that forest and you run out of water, it's going to have the exact same effect. It does not matter, these abiotic factors, on if it's a high density or a low density. It doesn't matter if it's a high populated area or a low populated area. It's going to affect it the same no matter what. So you're going to have your weather events that are going to be big here, such as floods or hurricane, tornado. It doesn't matter how dense the population is, a tornado is still going to have an effect on it because those do not relate at all, versus competition disease have a direct relate with the population and size. So just a couple more examples. The competition, we already went over that. That's going to be the density dependent, and then the disease, which you've seen play out with all of the COVID. These are the other examples of your density independent. I already talked about the tornado and if you ran out of raw water, which would be the drought down here, but also forest fires. This is one thing that you've probably seen a lot lately with the horrible forest fires that have been going on. But this forest fire, again, let's go back to the poor little squirrel example, but it doesn't matter how many squirrels are in this fire, in the forest, this fire is going to have the same effect on them, whether it was 100 squirrels or 2 squirrels. It doesn't matter. That's why it's density independent. It affects them independent or regardless of the density. So I hope that makes sense. I know I kind of rambled on a lot. If it does not make sense, please contact me so we can make sure we clear it up. Dispersion. This is our third feature. This is talking about how they're spaced out. So there are a few shapes that we can look at. It's going to be clumped, uniform, or random. When you're looking at all the individuals in a population, sometimes they all clump together, sometimes they're very uniformly spaced, and sometimes they're randomly spaced out. And so we're going to look at this in the next slide, and I'll draw you out a little bit of an example. So I'm going to go ahead and change slides. When you have random, then you are going to have species that are, or the individuals that you may have a little cluster here, and then they'll be really spaced out, and then they're kind of spaced out, and maybe another little cluster. There's no rhyme or reason to where they are. And then when you have uniform, they're going to be very evenly dispersed everywhere you go. You could almost measure exactly from where one species is to another. And then clumped is going to be like you have a cluster over here, and then a cluster over here, and maybe a cluster over here. So they all clump or they cluster together. So those are kind of pictures just to show you what your random versus uniform versus clumped dispersion is going to look at. And so take a second and think, what do you think would be examples that will go under each one? And a species that you think would have a very random dispersion versus a uniform versus a clumped. So think about that for a second, and then I'll give you a couple examples. Well, things like fish, because you have school of fish, or anything that travels in a pack like a wolf pack, they're going to be very clumped because they live together. You have penguins, which actually tend to be in a very uniform, spaced out area, which is kind of interesting. And then plants can be very random because the seeds will be picked up by the wind, and they'll just blow and go randomly down onto the ground. Or you could have animals that eat the fruit of a plant, and then as they're walking throughout the forest, wherever they decide to do their business, that's going to fertilize whatever seed they ate, and then it will grow. So you can see a lot of different very random patterns when you look at the populations of a plant. This is really just a graphic kind of explaining the things that we've already talked about that affect the growth rate of your population. So immigration is adding to the population, and birth rate is adding to the population. But at the same time, you have to take into consideration how many individuals are dying out of the population, because that's going to subtract, and then also emigrating, exiting, or leaving the population. And that could be because there's a lack of a resource, such as a lack of food, lack of water. So they have to emigrate or leave to find something else, or the individuals could end up starving. So that will affect this population size of that area. So how are populations measured? When we talk about the growth rate of the population, first of all, you must be thinking, well, how do we even know how many squirrels live in that forest, or how many zebra are within that savanna? Well, they do have multiple different ways that you can count and estimate what that population size is. One way you can find out what the population is to simply count the number of individuals. That's what we did in the picture that had the swans. There were only a couple. They're very easy to see. So 1, 2, done. You counted them. But when you're looking at the populations of, for instance, how many hammerhead sharks live within the ocean, that's not something that you can simply count. You're going to have to do an estimate by sample. And there's lots of different sampling things that scientists can do. But the one thing we're going to look at is something that we call mark and recapture. Basically, and I'll use these snails as an example, you, as a scientist or an ecologist, you would go into an area and you would capture some of those individuals of that particular species. And then you would mark them, just like the name says. So with these little snails, they put something on their shells that don't hurt the snail, and they have a little number that you can't read right here, but you can see where that number is. They have a number on each shell that is a specific identifier for that individual snail. Then they'd put the snails back out into the wild, and then they would capture some more. And they would look to see how many of those snails had they previously caught. If they throw the snails back, and when every time they make a new catch, they are capturing brand new snails, then that's showing them the population is going to be pretty large, because every time they throw them back, they keep getting brand new unmarked ones. But if they throw them back and they keep catching the same snails, then the population is pretty small, because that's why they're having to recapture the same thing. And you see that happen with like the hammerheads. They put the marker on their fin, and then you see them with the birds. They have it on their ankle. And this is a good indicator because it shows the health of ecosystems. Remember, in science, everything is connected. Sharks actually are a very important, vital part of the ocean ecosystem, and they are directly affected by coral reefs, and coral reefs are directly affected by them. And that's really a keystone species within our entire ocean ecosystem. So it's a very big deal that you have the health of them. And so scientists will measure the populations. Are they growing? Are they falling? What's causing this fall in populations? There's usually trackers so they can see where they're moving, emigrating, or immigrating into, all of that stuff. So just one more example of this. In this example, they had some fish. So the fisherman caught the fish. He marked them, threw them back. You can tell which ones are marked because they're colored in. And then he caught some more, and you can see that he has two, one here and one here, that he's recapturing. And so after you take many samples, you do this a bunch of times, a very large sample size, then you could use this formula to estimate the size of the population, and you can see that here. We're not actually going to do this, so you don't need to know the formula, but I just wanted you to know that that's what you would end up doing, is use that formula in order to find out an estimated version of your population. And this actually is very correct if you do a large enough sample size. In ecology, we had to do it with beans, for example, and the instructors knew how many beans were in each bag, and we would take some out and we'd mark them with the Sharpie, put them back, mix it, take some out, put them back, mark it, and we would do that for a certain amount of samples, plug it into our equation, and then we would get a number. And then we would have to count how many beans were actually in the bag and compare. And every time our estimated sample size was very close to the exact sample size that was in the bag. So math does work. And whoever came up with that formula is very smart. Last thing to talk to you about is an age structure. Now these are important to understand, so I need you to pay attention. You're about to do an assignment where you're going to have to answer some questions based off of an age structure that you see. And so I just want to show you how the age structures are set up. On this side, you have males, and this side, you have females. So when we're looking at the population, we want to look at how many people are, how many men are being born into a population versus how many women are being born into a population. And because of statistics, that's always going to be about a 50-50 percentage. So in the middle, or usually in the middle, they'll have your age group set up. Each graph may look slightly different, so you got to make sure you pay attention. Age group, you've got men, you've got women. Then going along this bottom line, this is showing you the population growth. And notice it says in millions. So this represents 4 million. This is 1.6 million, 3.2 million. So if I ask you a question and I say about how many men in Germany were of the age of 30 years old in 2016. Well, you'd come to the man side where you see the age range 30 to 34. And so you would just follow this line out, and I'd go down, and I'd say there's about, we'll just say about 2.5 million men lived in Germany in 2016 that were between the ages of 30 and 34. If I want to say, well, how many women lived in Germany that were about the age 95 to 99? Well, I'd come up here to that age, and I'd follow this down, and oops, not a very straight line, but we'll say, let's see, if that was, I don't know, we'll do about 0.3 million women were about the age of 99. So that's kind of what we're looking at when we have our age structures. It's simply a bar graph that's turned on the side. It's 2 bar graphs because one for women, one for men, and you just have to make sure that you're reading your axis, reading the information so that you can answer the questions off of it. I do have one last age structure I want to show you. Going back to this previous slide, if you look at this one for Kenya, oops, let me get my marker. You have this one showing the Kenya growth rate, and you have this one showing Italy. Notice this one says 0 growth, and this one says rapid growth. And the reason you can look at this and the U.S. is showing slow growth is because if you have a lot of people down here in the young age, you got a lot of people being born, so birth rate looks high, notice your death rate looks pretty low because you don't have that many people here in the area that would be dying. When you look at Italy, your number of the people being born and also looking at how many people are about the birth range, could give birth, your birth rate and death rate look to be about equal. So it's about a no growth, and this is going to be fast growth because this birth rate is so much higher than this death rate. And this birth rate's a little bit higher than the death rate, which is why it's slow growth versus no growth versus fast growth, which is what rapid is. So go ahead and work on your age structure assignment. Let me know if you have any questions, and I will talk to you later.